Hello, my name is Julian Miller, and welcome to the A-Life 2020 tutorial on Cartesian Genetic Programming, or CGP. CGP is a form of genetic or automatic programming which uses evolutionary algorithms to find solutions to computational problems. It can be applied to a large range of problems. It can even be applied to generating art. The two pictures on this slide were evolved with CGP. Here is a brief outline of the tutorial. We begin with a presentation on classic CGP, which is still widely used. In section 2, we look at various forms of CGP which have increased its efficiency or generality. In section 3, we discuss 5 applications which demonstrate the diversity of domains that CGP has been applied to. These are digital circuit design, object recognition, neural networks, artificial organisms, and artificial intelligence. In section 4, we discuss open source software for CGP and future work. Although the origins of GP are much earlier than 1992, GP became much more widely known after the publication of John Kozer's book in that year. As you can see, the 1990s was a period of much activity on different ways of representing and evolving computational solutions. In that decade, the major dialects of GP were invented. Kozer's tree-based GP originally expressed programs in Lisp, which can be represented as a tree. In 1996, Lee Spector described a stack-based form of GP known as push GP. Also in 1996, linear GP was described by Peter Nordin and Wolfgang Banzhaf. Linear GP is register-based and operates a bit like machine code. An early form of CGP was first described in 1997. And grammatical evolution was devised by Conor Ryan in 1998 and evolves programs defined in a user-chosen grammar. Although CGP grew from the work on evolving digital circuits in 1997, the term Cartesian GP first appeared at the Gecko conference in 1999. The term Cartesian arose because CGP was first applied and conceived for evolving circuits on field programmable gate arrays, which use a Cartesian grid of programmable logic blocks. CGP encodes a graph using a two-dimensional grid of computational nodes. The user decides how many rows and columns of nodes will be used. These are the rows, and these are the columns. The user also decides how columns of nodes should be connected. This is done using another user-defined parameter called levels back. Levels back defines how many columns of nodes on the left of a column of nodes may have their outputs connected to it. For instance, if levels back equals 1, then the nodes in a column can only have their inputs connected to the outputs of nodes in the preceding column, or the primary program inputs. So if you're on levels back equals 1, for instance, these nodes can only connect to the column of nodes immediately preceding them, but they're allowed to connect to the program inputs. The primary inputs and the outputs of nodes are all sequentially labeled or addressed. So the inputs are labeled 0 to n-1, and then the nodes are labeled immediately after that and sequentially going down the columns. Each node has a number of inputs, which are addresses. Let's look at an example. In this example, there are 4 columns of nodes and 1 row. So we've just got 4 nodes here and it's 1 row. We assume that the problem we're trying to solve has 2 inputs, these, which we've denoted X and Y, and 3 outputs denoted OA, OB, and OC. The addresses of the inputs are 0 and 1. And the addresses of the nodes are 2, 3, 4, and 5. The node functions are add, subtract, multiply, or divide, protected division, that is. And in the genotype, I've just underlined those. So underlined genes represent a function gene. So to obtain the phenotype from the genotype, we look at the outputs first. So let's look at OA. you can see that it's directly connected to the first input. So that's an easy one. So OA effectively represents the equation OA equals X. Let's look at OB. OB is connected to the third node, whose address is 4, which is a multiply node. And so we've got to look at its inputs and trace those back. So its first input is connected to the output of the node with address 2. So let's have a look at that. Well, the node with address 2 is connected to the 2 inputs, and it just adds them together. So the output of this node is y plus x. The second input of the node with address 4 is directly connected to the y input. It then multiplies those two. So you can see that the equation for OB is y plus x times y. And decoding OC is a similar, goes in a similar way. One thing to point out, you might have noticed this, that this particular node, the one with address 3 in a slightly different color, isn't actually connected to anything. There's no nodes on its right that connect to it. Sorry. This is a non-coding node. And we're going to talk quite a lot about that in subsequent slides. In a CGP genotype, many nodes Sorry, I got a bit lost with my bits of paper there. We just saw a non-coding node in the previous example. When the number of nodes is much larger, there are many non-coding nodes. What this means is if the genes of a non-coding node are altered, it makes no difference to the phenotype or the fitness, because they're just ignored. Let's look at a small example of how redundant is this genotype to phenotype map in CGP. Well, imagine you've got a genotype with only 9 nodes. That's very, very small. And you say, how many 3-node phenotypes could I possibly encode in a 9-node genotype? You'll find that there's 118,124 different genotypes, all representing exactly the same 3-node phenotype. We're going to look at how you decode a CGP genotype. But basically the pseudocode for this. You'll see it fits on this slide, and it's relatively straightforward. Step 1, we need to define a few arrays. A Boolean array called to evaluate, this one here, and a node output array which stores the outputs of the nodes and what the input values are. 2 arrays, graph input and graph output. These are defined by the problem you're trying to solve. An array to hold which nodes are actually used, the active nodes. And of course, we need some arrays to hold the genotype itself. And I've got an output gene node, which holds the output genes. And I've got an array of nodes, which, as you'll see, has connections within it. So the next step is to identify which initial nodes need to be evaluated. Well, these are the nodes that are pointed to by the output genes. So you can see that you have to immediately set in the Boolean array to evaluate, set those nodes to be true, because you definitely need those. Once you've done that, you can then go through the to evaluate array, working backwards and working out what other nodes you should evaluate. And as you do that, you set these, the to evaluate Boolean array to be true, telling you where to go next. And while you do that, you can record which nodes are actually used along the way. Step 3, We're now ready to load up the input data values. So the first thing that happens is that the graph inputs, which are supplied by the type of problem you're trying to solve, are written into this node output array. Step 4, now we can execute the graph. We know what nodes are used, so we can get the inputs to each node, carry out the function defined in the genotype, and actually compute what the output is and write that back into the node output array. Final step is to get the outputs from the graph. So you just go through all the output genes and look at what the value is of node output at those points, and those will be the outputs from the CGP graph. CGP uses a very simple evolutionary algorithm called a 1 plus lambda evolutionary algorithm. This has a population size of 1 plus lambda. Lambda is most often chosen to be 4. So in other words, the population size is actually 5. So the first step is you start by generating a population of 5 randomly generated genotypes. And I assume that's been done. And then you have to find the fittest genotype, and you call that the parent. So here we can see that this genotype has fitness 10. I'm assuming we're trying to maximize the fitness. So this is the parent. This gets promoted immediately to the next generation. And then the other members of that population are mutated versions of the genotype, which has fitness 10. Now there's an important step that you have to observe here, and that is that sometimes when you work out what the offspring of this genotype is, you find that there's another which has a fitness that's the same as the parent, but not better. In that case, you always choose the offspring as the new parent. And we'll go into detail why this is important in subsequent slides. But it's worth noting that often offspring have the same phenotype as the parent because they only differ in non-coding genes. And of course, you could detect that in advance, and you avoid calling the fitness function again because you already know its fitness. CGP mainly uses a very simple mutation operator called point mutation. And this, first of all, the user decides how many genes should be mutated. You choose a gene at random here, and then you interrogate it to find out what kind of gene is it. Is it a connection? Is it a function gene? Is it an output gene? And then, depending on what type of gene, you change its value to a new allowed value. You can also do this probabilistically, but this would mean walking along the genotype and deciding whether you should change that gene or not. Point mutation is very efficient because you only go to a small number of locations where you're going to change the genes. In 2003, Brian Goldman and Bill Punch developed a new form of mutation called single, which basically means you just mutate one gene at a time, point mutation, until you hit an active gene. The interesting thing about this form of mutation is you don't need a mutation rate parameter because you're just going to mutate a gene at a time until hitting active, where you stop. So this requires very little change to the point mutation process, which we looked at in the previous slide. So you can see all you need to do is detect whether the gene is active and have a loop that keeps mutating until you hit an active gene. This graph shows the results of an experiment done by Goldman and Punch in their paper in 2013. On the y-axis, we see the median number of evaluations. We see the median number of evaluations until success. On the x-axis, we see different mutation rates. And what's being plotted here is how the median number of evaluations until success varies with mutation rate. So the blue line here is standard CGP, and you can see that you'd have to do a lot of experiments to find out what the ideal mutation rate is for standard CGP. This is this point here. By the way, these results were compiled on a problem called the 3-bit parallel multiplier, which is quite a difficult circuit synthesis problem. Now we can look at how this number of evaluations varies with on a slightly different strategy, which we call SKIP, which is this green dotted dashed line. And in that, we don't increment the evaluation pointer if the fitness of the genotype is the same as the parent. So you can see that radically affects the graph of how the median evaluations for success varies with mutation rate. This line here is the single strategy we just discussed. You see, it doesn't have a mutation rate, so this is why it's just a line here, and you can see that it's considerably better than the other approaches. There is another mutation strategy that they discussed, and it's called accumulate, where you mutate multiple genes until some active genes are changed. In 2015, Goldman and Punch observed that there was a considerable positional bias in CGP. That is to say that the active nodes are much more likely to appear on the left near the inputs. There's an obvious reason for this, because the leftmost node can be connected to any of its nodes on its right. That's almost all the nodes. And as you move along the genotype, the number of nodes that you could connect to that output decreases. So when you look at CGP genotypes, you'll find that most of the active nodes are bunched around the left-hand end of the genotype. So they thought, well, suppose we reorder the nodes prior to mutation. So we redistribute the active nodes, we move them out throughout the genotype to try to remove that positional bias. And they found that this produced better results than standard CGP, especially on harder problems. So we're going to return to this issue much later on, where I'll make a suggestion about an interesting way of doing this that hasn't been tried yet. Calcroit recently examined the utility of two new mutation operators, which he called insertion and deletion, which are, they know about whether nodes are active or inactive. So the insertion mutation mechanism chooses an inactive node and changes one or more connection genes in the genotype to make it active, thereby inserting a node in the genotype. Deletion does the opposite. It alters the connections to an active node so that the node becomes inactive. And he examined the impact of these operators together with point mutation and found that the combination was more effective than point mutation alone. He did this on 3 Boolean benchmarks and a suite of symbolic regression problems. But obviously, we need more detailed studies on a wider range of problems to evaluate these operators. People often ask, why don't you use crossover in CGP? Well, back in 1999, when I first proposed CGP, I used a standard genetic algorithm with crossover. However, it proved to make no difference to the results. For certain kinds of problems, which are multiple output, and you get those a lot in circuit design, you could devote a chromosome to each output, rather than having one genotype with multiple outputs, which is what I've described before. You have multiple chromosomes, each with a single output. Now, sometimes you can be in a position where you could evaluate the fitness of each of those separate chromosomes independently of the others. In that scenario, which was investigated by Walker and Miller in 2006 onwards, you find that crossover can be very, very effective. Janet Clegg and others in 2007 developed a slightly different representation of CGP in which all the genes are floating point. And she used crossover in this representation and found that on symbolic regression problems, crossover worked very, very well. However, a little bit later, a master's student at the University of York, Andy Turner, did his master's project on this representation and discovered that sometimes crossover worked better in that representation and sometimes worse. So the bottom line there is that jury is still out about whether this floating point representation is really useful for crossover. There's been a couple of other attempts at creating crossover mechanisms in CGP. Paul Kaufman and Roman Kalkreut have both devised some. Kalkreut has investigated subgraph crossover, where active nodes are swapped either side of a single point crossover point. But nothing definitive has been found about the utility of crossover. So I guess it's still an open question about whether there is a form of crossover that makes CGP much more effective. Neutral search is fundamental to CGP. So when mutation does not cause a change in fitness, genotypes can drift in the genus space. This is actually called neutral drift. Indeed, neutral drift is recognized as important in biological evolution, principally through the work of Motu Kimura. And over the years, there have been various studies showing that neutral search is extremely important to CGP. The graph in this slide shows this very well. It compares the final fitness obtained in 100 evolutionary runs when offspring can only replace parents if they have a better fitness, compared with choosing offspring over the parent if they have the same fitness, which is standard CGP. So you can see that the blue results are standard CGP, and most of them nearly either solve this problem perfectly or come very close to solving it. The other points here are where you only accept a genotype to replace a parent if it has a better fitness, not equal. And you can see that in those 100 experiments, it wasn't able to solve this problem once. So it's quite compelling evidence. How big should the genotype be? Well, we've seen that neutral drift is very, very important in CGP. So you might think, well, if I have a lot more redundant nodes, my search should get more and more efficient. So this was the motivation for a study by Miller and Smith in 2006, where they looked at the efficiency of CGP search as a function of the number of available nodes and mutation rate. And they did this on a couple of small circuit problems. They found to their surprise that the least number of evaluations was required for success occurred when 95% of the genes were inactive. And also that the efficiency of the search appeared to continuously improve as the number of nodes increased. Well, of course, this can't continue because it would suggest that if you kept increasing the genotype, eventually you'd solve your problem in the initial population, which cannot be true. So it required a more detailed study to look at this, and this was done by myself and Andy Turner. And we looked at programs going up to sizes of 100,000 nodes, and we looked at 4 different problems to see whether there was an ideal size for the genotype. So here are the results. So this shows you the results on the double pole balancing problem. We are trying to balance 2 poles on a movable cart. We're trying to minimize the error here. And what we're looking at is how that average error varies with the number of available nodes that we've made available. So just concentrate on the blue lines for now. So you can see the first thing you can spot when you look at all four of them is that they all have a dip in the middle, some more pronounced than others. So when you look at the ideal number of nodes to use in these four problems, you find that double pole is best with about 50 nodes. Whoops, sorry. It's best with about 50 nodes. The 4-bit multiplier, which is a very difficult circuit synthesis problem, seems to perform best when the genotype has 100 nodes. 8-bit parity best seems to happen around 600 nodes. And on a very, very difficult classification problem known as the tower problem, you find that the ideal number of nodes is around about 5,000. So you can see it varies a lot, but there's always some ideal number of nodes. So we're going to look at, in the next section, different forms of CGP. So far, the CGP nodes have all been specific functions defined in the function set. Modular CGP, or sometimes called embedded CGP, allows CGP programs to be captured and reused as new functions in the function set. We call such functions modules. John Koser introduced such a concept in genetic programming, and he called them automatically defined functions. So in this form of CGP, you can capture modules. They're dynamically created or destroyed. They can be evolved, or they can be reused. Let's look at a conceptual animation showing the basic principles of this. We assume that only one row CGP is being used. It's only being devised for one row CGP. So here's your genotype, just a string of nodes, CGP nodes, and we are going to create a module. This is the first thing we're going to do in this animation. So module creation generates two random points in the CGP list of nodes above and extracts all nodes between these points and uses them to define a new subfunction which we add to the list of modules. So those are the nodes being captured, and they are added to the module list, and a new function, a new node function, is put in the genotype. When we replace the nodes extracted with a new subfunction, we give it a special type. That's why I've drawn a triangle there. Basically, that means it was captured via module creation. I'll talk a little bit more about that in a second, but it's quite important. How do we reuse a module? Well, this happens by mutation. So mutations might occur and change primitive function nodes into modules. So I've indicated that here, where I've put I've made the nodes diamond shapes. The reason why we do that is we have to keep a record of the types of the nodes. So these diamond shapes represent the same function as the triangular node, but the shape just indicates that we know how it arose, that it came into existence because of mutation, rather than module capture. Of course, when you capture modules, we've talked about using two random cut points. They're going to be random modules, so they're not going to be particularly good modules. So you need a way of allowing evolution to get rid of modules as well. This is called module destruction. So module destruction only operates on a module that's been captured in the first place. Hence, it acts on that triangular node, which was a module. Basically, it takes the module out and inserts its original code back in. You can see after that, it still leaves the two calls to that module that were created through mutation. Module evolution occurs when we mutate the modules, we mutate the code inside the modules that are on the module list on a particular module. So I've indicated that here. Now to come back to these different types, which might seem a little confusing, by using the types, we can track how modules occurred in the code, and you can prevent a phenomenon called bloat this way. In standard GP, programs grow over evolutionary time, and that's called bloat. But through the use of module types, we can avoid that in modular CGP. Now, the big question is, how effective is modular CGP when compared with standard CGP? So is it more effective or not? And the way you should do this comparison is you should make sure that the total number of primitive nodes available to both approaches is the same. So the thing is that with modular CGP, it's got modules, and each of those modules has got primitive nodes in them. So you need to add up all those primitive nodes and set that as that has to be the same number of primitive nodes as used in CGP when you do your comparisons between the two. And I'm sorry to say that rigorous comparison wasn't done in the IEEE transactions paper on modular CGP. So it actually was an unfair comparison. So what happens if you do a fair comparison between the two? Well, here you can see two different experiments comparing CGP with modular CGP on a number of parity problems. So as the number of inputs to the parity grows, the problem gets a lot harder. And you can see that when you only allow 100 nodes maximum, these are primitive nodes in CGP, or the sum of all the nodes used in modular CGP, you find that modular CGP starts to work better for hard parity problems. If you look on the other side, now we've increased the limit of nodes to be 500 nodes, and you do the same comparison, you find that actually standard CGP works as well on this sequence of parity problems as modular CGP. So there doesn't, at present, it doesn't look like having modules gives you much of an advantage. So my view about this is that modular CGP is a really nice idea, and it ought to help a lot, but it doesn't seem to help over standard CGP. So the question is why, and how do we make sure it does help? I think this is an open research question. Now we're going to briefly look at another form of CGP, which is basically standard CGP, but it allows recurrency. So here we relax that restriction. In standard CGP, up until now, we've assumed that all the inputs to nodes must come from nodes on the left or primary inputs. So recurrent CGP relaxes this so that nodes can receive their inputs from a node on the right. So you can see a case of that here is this node is getting its input from a node on the right. This is relatively easy to do in CGP. You just need to take a few precautions. You need to think about how to prevent indefinite recursion. That means that you keep having to process recursive nodes until some limit. So to prevent that, you initialize all active nodes to output 0. You apply your inputs, and you update all the active nodes once from the program inputs to the program outputs. So you only update them once, and then you read your program outputs. So it's a kind of restricted recursion, which prevents this indefinite recursion. Recurrent CGP also has a probability of a connection being recursive, a controllable probability. And we call that recurrent connection probability. So you can actually choose to have graphs with only a certain level of recurrency. This form of CGP was applied to a couple of problems: artificial ant trail, series prediction problems, and mathematical sequence induction. And it found results that compare very well compared with other methods. CGP can quite easily encode algorithms. The way you do this is you kind of create a nominal CGP. So I'm imagining here that each of these nodes carries out quite a complicated operation, which is defined somewhere else. And basically, you just use CGP to give you a combination of operations, which effectively represents an algorithm. So you're making use of the fact that CGP has non-coding nodes, which can be ignored, and so you'll get a variable length list of operations, which you can apply to some sort of data structure, and it can represent basically an algorithm. A development of this type of CGP was developed by Patricia Reiser Welch in 2016, and she relaxed the acyclic nature of CGP. So far, apart from the recurrent CGP, CGP I've described it as being acyclic. In other words, nodes can only connect to nodes on their left. So she relaxed that so that you could create cyclic graphs. And she applied this technique to a number of real-world problems, traveling salesman problems, nurse rostering, difficult real-world problems. To do this, she had to create a number of extra genes that we don't have in standard CGP, branching genes, which define loops inside a graph, and condition genes, which encode a stopping criterion for this loop. And you have to, you can easily get nonsensical loops. So you need to modify the mutation operator so that only accepts valid loops. So here's the genotype. It's got a lot of extra genes because it's got these branching genes and condition genes. The other genes are standard CGP. You can see here it encodes a graph with loops. When the loop's finished, it jumps to the next instruction outside the loop. Now we're going to look at a very interesting different form of CGP called self-modifying Cartesian genetic programming. This was inspired a little by biological development. I mean, in biological organisms, brains are built through a process of development. Neurons grow and new neurons are born from those neurons, and it creates this much larger structure. So I like to think of it as inspired by biological development. How does it develop? Well, it includes self-modifying functions in addition to computational functions. So the function set has become bigger, but some of the extra functions, actually what they do is they don't really do anything computational. They just pass through their first input or something like that. But when you obey them, they modify the graph itself. So they might say, take these 3 nodes and move them to some other place in the CGP genotype in the graph, or they might say delete some nodes. So they actually alter the code itself. It's a general purpose GP system, so you can apply SMCGP to any of the problems that standard GP can be applied to. And in fact, you can apply it to a lot more problems than you could have conceived of applying standard GP to. So the interesting thing about this form of CGP is that when you apply these self-modifying operations, you get a new intermediate phenotype, and you can say, does this intermediate phenotype solve this particular problem? Then you can apply some more self-modifying operations and get a new intermediate phenotype, which might actually solve another problem. So in principle, you can solve an infinite number of problems using self-modifying CGP. Does it always self-modify? Well, no, not necessarily, because if it's not useful to self-modify and create another intermediate phenotype, it can actually switch off its own self-modification, because the code could change itself in such a way that it has no self-modifying functions in it, in which case it would become like standard CGP. In order to create self-modifying CGP, it was necessary, because we're going to be solving multiple problems, it was necessary to make some representational changes from classic CGP. The first thing that we got rid of was absolute addressing. In standard CGP, connections to nodes represent an absolute address in some data, in an array of data. We saw that when we did the decoding pseudocode earlier on CGP. In SMCGP, we assume that all the connections are relative. And by the way, SMCGP, or at least the version I'm talking about, only applies to one-dimensional CGP, where there's only one row. So you count back to work out where you're going to get data from a particular node. You count back nodes from the node you're on. And that's what we mean by relative addressing. The next thing that we changed is we got rid of absolute inputs, if you like. So now we have we have additional functions in the function set, which we call input gathering. And we've got 3 of these, and they work a bit differently. What they do is they work with a global internal pointer. So you imagine you've got a register of your inputs, and for instance, this function, the input function, will return the value in that register, that input register. It will return that value. But after it's been called, it advances the pointer in the register by 1 place. It increments it. And note that this register of inputs, which is just your problem inputs, is assumed to be circular. So if you keep incrementing the pointer and you go beyond the register, you come back and read the first entry in the input register. The function input p is the same as the input function, except that it decrements the pointer. And skip imp takes one of the parameters, and we'll come on to this in a minute. Self-modifying operations have parameters. It takes one of the parameters and it moves this input pointer by more than 1 place. Often in CGP, you can get a situation where you get inputs to nodes that are count back beyond the length of the graph. So you need to handle this case. When that happens, we just assume that they get the value 0. The next change is outputs. We want to be able to handle problems with multiple outputs. Each problem might not have the same number of outputs. How can we do that? Well, we've got rid of output genes, which is what you have in standard CGP. and we have now output producing functions. So we have a function in the function set which says, I'm an output, I'm going to return the value I'm given. So that way, mutation can insert outputs, and you can supply as many outputs as you want. You need to set up quite a few defaults about what to do in various situations, because you could have an intermediate phenotype in CGP which didn't happen to have any output functions. So what would it output then? So in that situation, you have a default, and you just say, well, we'll take the output from the last active node used. And you might need other defaults in situations, for instance, where the number of outputs does not match the number you require. So it's a little more complicated, but perfectly doable to do that. So I mentioned, for instance, in the skip input function, that it uses a parameter. We actually call those arguments. The self-modifying operations, as we'll see shortly, they alter the code, so they need arguments to tell them how they operate, where they start, where they stop, and so on. So all nodes in SMCGP have arguments, whether they're self-modifying or not. If they're not self-modifying, they just ignore their arguments, because they just take data inputs and they process them and provide an output. They don't need arguments. But if they're self-modifying, they will use those arguments. And these arguments are 3 floating point numbers. Now, most of these self-modifying instructions, if not all, require arguments that are integers. And so in that particular case, the floating point arguments would be cast integers. So here's a brief summary. In SMCGP, every node has a function type, like standard CGP. It has connection genes, but these are all relative addresses. They count back from the node you're on. And it has floating point numbers, which may be used by self-modifying operations, in which case they would be truncated to integers. So in SMCGP, as we've discussed, there are 2 types of functions. There are computational functions, the usual standard CGP computational functions, and there are also self-modifying primitive functions, which are only passive computationally. They just typically pass through their first input, don't do anything to it. But if they're self-modifying, they use their parameters, and here's a small list for you showing you some of the functions you might have. So you might have a self-modifying instruction called move, which moves the nodes between the start argument and the end argument. Basically takes those nodes out and moves them into the position specified by the insert argument. And you might have delete operation, which deletes the nodes between start and end. So it doesn't have to use all the parameters, it depends on the function. Here's one called change function, which changes the function of a specific node, a specified node, to a specified function. Actually, the number of self-modifying operations we tend to use is reasonably large, because there's many things you could do to a graph to modify it. How do you execute a CGP genotype? Well, the first thing you need to do is you duplicate the genotype to what I call an intermediate phenotype. And it's the intermediate phenotypes that you execute. So by execute, I mean apply the self-modifying operations to it to see what new graph you get. Then you would evaluate that new graph. You feed in inputs and look at the outputs and see if it solves your problem. Then after that, you'd apply some more self-modifying operations, get a new graph, and check whether that new graph works well on a different problem. So I've more or less said that programs are iterated, so you get a whole sequence of these intermediate phenotypes, each which may solve a different problem. Now, how do you add self-modifying instructions? Well, we have a process called triggered. I'll come on to that in the next slide. But the other thing is that you have a to-do list, so you don't actually have to obey all the self-modifying instructions in an intermediate phenotype. You might decide that my to-do list has only got 3 instructions. I'm only going to obey the first 3. So you start at the left-hand end of the intermediate phenotype, and you apply a self-modifying instruction that you encounter that's active. Then you move on to the next one, and then you might stop when you've reached your to-do list, and then evaluate the intermediate phenotype. This is nice because you can control how much self-modification happens. So how do you add functions to the to-do list? Well, we've talked about triggering. So the default is if they're active, then you add them to the to-do list, unless the to-do list is full, in which case they're ignored. But you could also allow it to interrogate its inputs. So you could say, is the actual data being presented at the first input bigger than the data being presented at the second input? In which case, we will allow that self-modification, that active self-modification, to be added to the to-do list. Okay, so we've talked a lot about how SMCGP works. Let's look at an example of it operating and solving a problem. Now here, the problem we're trying to solve is even parity. I don't mean a particular even parity, I mean all even parity. So we're aiming to find a solution that can produce all parities just by iteration. So that a general solution. So what you wanted to do is, on the first iteration, you want your intermediate phenotypes to solve 2-bit parity, which of course is very, very simple. And you've got the solution to that as one function in your function set. But on the next iteration, when you apply the self-modifying operations and generate the next intermediate phenotype, you want it to solve 3-bit parity, and then when the next iteration 4-bit, all the way up to 22-bit parity. The aim being to find the general solution. The hope is that at some point, suddenly, the intermediate phenotypes will automatically generate the next parity function. The fitness was the cumulative sum of incorrect bits, so we want to minimize it. We want perfect solutions. And as I said, the aim is to find a general solution. And in actual fact, we did that. We found general solutions, and there's a mathematical proof that is the general solution provided in our GPEM 2010 paper. It's worth noting that both CGP or forms of GP cannot solve this problem directly, as they all have a finite set of inputs. So with SMCGP, you can find general solutions to problems. This is a very interesting graph. It shows you how many evaluations it took to solve a particular parity problem as a function of the number of inputs to the parity. And it showed, this is an evolutionary run, and it shows you that the number of evaluations growing as the problem gets harder, as you're trying to solve more and more parity problems, 2 parity, 3 parity, 4 parity, and so on. And then at a certain point, around about 6 parity, you find that no more evaluations need to be done. So evolution suddenly, when you iterate it, produces all the other parity. This kind of figure is what you need to see when you're looking for generality. So it looks like suddenly, for free, without any more evolution, it produced all the remaining parity functions. And this was indeed the case when you analyzed the program. Another problem we looked at with SMCGP was evolving pi. So can we evolve an algorithm that will compute pi to greater and greater precision? So here we allow 10 intermediate phenotypes. We iterate a maximum of 10 times. If the program output does not get close to pi at the next iteration, the program is stopped and we add a large fitness penalty. And the fitness at iteration I is the absolute difference of the output at iteration I and pi. So we're trying to minimize the absolute difference. We allowed one numeric constant to the SMCGP graph. And what we found, this was one evolved solution, a particularly interesting one, is that when you, well, the first thing you can see is we've evolved a solution that's fairly readable. It's a recurrence relation, and it has a strange condition to start it off at iteration 0, which is it just produces a number. I can't remember what number this is. Then after that, it uses this recurrence relation, and actually this expression can compute 2048 digits of pi after 10 iterations. So it's an extremely rapid approximation of pi. And in fact, in that same GPEM paper, I proved that this actually is the general solution. This converges to pi as I tends to infinity. Further results. Well, we've evolved a program that you can prove carries out bitwise addition of an arbitrary number of inputs. We've evolved a program that can compute E, Euler's constant E, to arbitrary precision. We've evolved functions that generate the first 10 Fibonacci numbers. I haven't analyzed them, but they probably were general. And various other functions have been evolved using SMCGP. One thing to note, you can apply SMCGP to a standard GP problem. It doesn't have to have, it doesn't have to be a sequence of problems. In other words, it doesn't have to need self-modifying functions. And when we did that, we applied it to a bioinformatics classification problem. Actually, SMCGP performed no worse than CGP. So that's good news, because it can actually solve general problems and multiple problems, but it can also solve a single problem. So in the next section, we're going to look at these 5 applications of CGP. The first application we're going to look at is digital circuit synthesis with CGP. Vasicek and Sekunina showed that you can actually optimize digital circuits with hundreds of variables using CGP. And actually they won the $3,000 Silver Award in the Human Competitive Workshop in Gecko 2011 with this work. Their method very cleverly employs a SAT solver to identify whether 2 circuits are logically equivalent. In many cases, you can do this in polynomial time. So this is how it works. Their method uses a traditional circuits optimization and synthesis tool to produce a reference circuit. And then they need to know whether an evolved circuit is logically equivalent to this reference circuit or not. If it is logically equivalent, then you say, well, we want it to have as few gates as possible. So they cleverly join the reference circuit and the evolved circuit in this arrangement with these exclusive OR gates and OR gate, so that it transforms the question of whether this and this circuit are logically equivalent. It transforms that into an instance of the satisfiability problem. So if the reference circuit and the evolved circuit are not functionally equivalent, then there is at least one assignment of the inputs for which the output of this circuit is 1. And then they can use their SAT solver to deal with that problem. So as I said, they begin they use a conventional state-of-the-art logical synthesis package to provide a seed circuit, which they encode into CGP. Then they apply CGP to a population of circuits, which includes the seed, and eventually they get an optimized circuit. The fitness function works as follows. You use the SAT solver to decide whether the candidate circuit, the evolved one, and the reference circuit are functionally equivalent, as I've mentioned. If they are functionally equivalent, and you say the fitness of the candidate circuit is the number of nodes minus the number of gates in the reference circuit. So you're trying to minimize the number of gates. The smaller the number of gates, the larger the fitness. If they're not functionally equivalent, then the fitness is, you just give a fitness of 0 to that circuit. And actually, they've made this circuit, this method much faster by using a circuit simulator, which they wrote prior to using the SAT solver to disprove the equivalence between the candidate circuit and its parent. So this is a bit of a breakthrough, and this group is still working very much on these problems, making it even faster. And actually, they're working on approximate circuits a lot these days. The second application is about evolving image filters with CGP. And this is work done by Simon Harding and others in 2013. So the aim is to detect and locate objects with some iCub cameras, which are on this cute little robot. And the principle of it is that you want to evolve a program that takes images and identifies the object of interest. So we call that the target. So you can see here, in this case, an example we're going to discuss trying to detect a particular packet of T. So there's a number of packets on this table. We want to be able to track a particular packet of T. So we're going to use CGP to work with images and produce this mask, which identifies whether pixels belong to the object of interest or not. So the input data is, for each image, you have a collection of images with objects on the table. And then you pass those through various filters, grayscale, red, green, blue, hue, saturation, luminosity for each image. So this is the input data for the CGP evolved program. Then you use CGP to evolve the filter to try to detect pixels of the target. Actually, Simon Harding used a genotype representation for this particular work, which was like SMCGP, but he didn't have any SM self-modifying functions in it. So in other words, he had relative addressing, as you can see here, function genes, and he has a floating point parameter. So it's interesting that some of the aspects of the SMCGP representation can be carried over to a slightly different form of CGP. He used a very large function set in this work, where there are many functions, and you can see some of the self-modifying types of functions. So he had input gathering functions, borrowed from the self-modifying work, but he also has some quite advanced image processing functions, which are standard domain knowledge in the field, various types of filters and so on. So he's making use of good domain knowledge here for this particular problem. So the fitness is the sum of the mean square error of pixel values between the image input and the target. So you can see a collection of images here and where the objects that we're interested in are located. So this is the CGP graph showing you some images coming in and undergoing various operations according to the primitive functions of the function set, and then producing this mask, which was the desired output. And actually, this is the program that CGP actually evolved. So you can see it's really quite small and reasonably readable. So this is one example of how this program operates on real-time video. So you can see it's Lipton's Andalusian tea that we're interested in. Notice how this image filter can detect any part of this object. So it did any side it detected it was part of this object, which is interesting. Actually, this is a fairly long little video, so I'm going to skip it, but actually it works quite well when you partly obscure the packet of tea, or you put it under different lighting conditions. It still can detect that. So the third application it uses CGP to encode artificial neural networks. So this is a natural fit for CGP because CGP encodes graph and neural networks are graphs. All you need to do is to augment the connections with weights. So you allow every connection gene to also carry a real numbered weight. This has been tried on a range of problems: pole balancing, ball throwing, classification, breast cancer detection, time series prediction. And it produces very competitive results with other methods in the field. This is a very interesting result. What Andy Turner did here was he looked at using conventional neuroevolution with a fixed grid of a number of nodes per layer, neural nodes per layer, and a number of layers. And he looked at the efficiency at evolving a solution under this condition for ball throwing, double pole, and the monk's problem, and notes that for these problems we want to maximize the fitness in these two cases, ball throwing and double pole, and we want to minimize the fitness for the monk's problem. So we did standard neuroevolution with a fixed topology using different numbers of nodes per layer and layers, and he compared that with using CGP encoded artificial neural networks using various rows and columns. The most interesting thing is that you can see that with CGP you get good results irrespective of the number of columns and rows. It's pretty uniform, so you get good results whatever the topology, whereas with conventional methods using a fixed grid, it's very, very dependent on the topology of that grid. So you can see that that's another benefit of using CGP to evolve neural networks. The fourth application I want to discuss is very much relating to A-life. I was interested in growing a self-repairing, self-maturing, cellular automata-based organism. You'll see what I mean in a minute. So what I made use of is something that CGP handles very well. It can handle multiple output programs very, very easily. So you can have a large number of outputs and it can handle that situation. So the idea of this work is to evolve a program inside a cell, which is a cell in a cellular automaton, that can construct a multicellular structure with various properties. So I put a single cell in a grid, cellular grid, and I run a CGP evolved program. And this program can read its own state. It's got discrete states, the states of its neighbors, and the amount of chemical at neighboring states. So I've got simulated chemicals as well. And cells can have a number of types, which I depict by different colors. And cells can change their type. They can replicate. When they replicate, they can actually grow over all 8 neighbors at once, which is not biologically plausible, but it's a fine assumption for A-life. Cells can also die, so they can, as it were, go into the death state Simulated chemicals naturally diffuse over the grid. So I've just drawn one picture here, but with chemicals you'd have several of these maps, one containing the cells and one each for each of the chemicals. So here's an outline of the program inputs and outputs. Actually, the program was all represented using binary operations. So all these inputs are binary. It's a large collection of inputs. I even have an environment input, so I can evolve programs that get the cell to do, the organism to do different things in different environments. I'm not going to show you those here. So I have streams of bits that are to do with the neighboring chemicals. And I have cell type bits, so I've got a maximum of 4 cell types. And so there's a stream of type bits for the types of neighboring cells. They're represented in CGP, and they output new chemical levels at the position of the cell, what the type of the cell is, and how the cell should grow. Should it grow or not? If it does grow, it grows into all these positions, provided that this signal is 1. So it can grow over a number of cells. So the first experiment was to try to evolve a program inside a cell that grows into a 63-cell organism that looks like the French flag. Why the French flag? I took that because in Lewis Wolpert's book on development, he talks about development from the point of view of trying to construct a French flag in a cellular way. So the fitness method, we compare the organism cell by cell with the predefined perfect French flag at these 4 iterations, consecutive iterations. So what I want is a program that takes one cell, which is a small element of this picture, and it grows, and it looks like a French flag at 7, and it looks like the same French flag at iteration 8, 9, and 10. So this is what I got. So this shows you the cells, and this shows you what's happening to the chemical. So you can see that it successfully, I'll run that again, it grew from a single cell, one cell there, and rapidly grew, and it stayed the same. And if you carry on running this program, it still looks like that. This is what the chemicals were doing at the same time as those cells were growing. So that was interesting. I evolved a program that produced this differentiated cellular structure, but it also has a maturity. It stops growing at a certain point. So the question is it doing anything interesting when it's in this state where it appears to be this French flag? Is it actually doing anything at further iterations? Well, you can investigate that by disrupting it. So what I can do is I can take the cellular map and then just make some alterations to it and then just continue to run the program. And what you find is immediately there's activity, and it reorganizes itself, and eventually it arrives at this slightly distorted French flag, and after that, after running the programs, it doesn't change again. So it's very interesting because it's learned how to repair itself and then shut itself down and reach maturity. Incidentally, the solution I showed you on the previous slide wasn't the fittest solution. It was the 3rd fittest solution. The other 2 solutions produced a slightly better French flag at the required iterations, but they didn't self-repair. This shows you what happens if I carry out surgery on the organism. I just remove the red and the green areas, the red and the blue areas, and I just run the program from that point. And again, it gradually develops, it gradually differentiates itself into the 3 different colors. And once it reaches that, there's no further change. So here's a few more behaviors of the maturing French flag when it's damaged. So let's look at what happens when I remove the white region. So I kill off all the cells in that region and then run the program from that point. Again, it reconstructs itself more or less and shuts itself down. What about if I swap the red and the blue regions? So I just change the state of these cells to red and I change the state of these to blue. We run that. That again differentiates itself, but actually it carries on growing. So if you like, a little bit cancerous, it just keeps on growing. This is an interesting one. What I did was I took the same number of cells as this has, and I changed their states and put them at random positions. So I just have this random blob of different cells. And I'm going to run the same program that produced this in that arrangement of cells. This shows you the chemicals. So this again sorts itself out and stops growing. So that's very interesting. Here's one where I've cut a big hole out of it, so I've killed off a lot of cells in the middle. And I think this one grows uncontrollably as well, but you see it differentiates itself back into these three regions. Yes, and it's got a little structure here that it just indefinitely grows, it can't get out of. So the fifth application I'm currently working on, and I have a couple of papers here at this A-Life conference on this subject, in the main conference and also in the Devo NN workshop, which I hope a lot of you will come along and see, virtually that is. What I'm trying to do here is to evolve neural programs. So I am evolving neural networks, but I evolve a program inside a neuron, two programs actually, which when they run, they construct the network. And the interesting thing about this approach is you can build a brain-like network that can solve multiple problems simultaneously. Because I'm interested in general problem solving. I don't want to just solve one problem. I want to find a way of evolving a brain-like structure that can solve an arbitrary number of problems, which is much more similar to the mammalian brain. So far, I've applied it to some standard benchmark problems in classification and reinforcement learning. So it can solve classification and reinforcement learning problems simultaneously. The same brain can solve multiple problems. So this is ongoing work, and you can learn more about that if you attend the other sessions in the A-Life conference. So we're going to stop there for the moment before we have a very short break, and then we're going to talk about further work and open source software. There are many CGP resources available online. First place to go for those is the CGP website, cartesiangp.com. You'll find there some C implementations of CGP and SMCGP that I've written. But I would recommend people who are new to CGP to use Andy Turner's package, which can be you can have a binding of that to many other languages, although it was written in C. It's highly extensible, easy to use. You need very few instructions to get into it, and it also encodes CGP ANNs. There's a Java implementation available by Eduardo Pedroni. Zdenek Vasicek has a highly optimized C machine code implementation. And of course, you can go to the CGP book for many articles on different applications of CGP. Dennis Wilson has implemented CGP in Julia fairly recently. There's another Java implementation of CGP by David Aranchak. Brian Goldman has implemented CGP in Python. Jordan Pollock in MATLAB. And Lawrence Ashmore has developed a Java evolutionary art package that you could use. CGP doesn't stand still. There are many people working on CGP all over the world, developing slightly different versions. Dario Izzo and his colleagues developed a form of CGP which they call differentiable CGP, in which all the functions are truncated Taylor expansions, and connections can also be weighted. And because through this, they were able to allow a form of back propagation to either adjust Taylor expansion constants or weights. And they applied this to very difficult symbolic regression problems with irrational constants like pi and e, and the solution of partial differential equations with excellent results. There's another form of, recently developed form of evolving graphs, a method of evolving graphs for GP, developed by Atkinson et al. I think that's worth looking at. It has it's slightly less constrained than CGP, so I think it might be worth looking at to find ways of making CGP even less constrained than it currently is. One thing I'd like to point out, GP is usually applied to a certain domain of problems, and that's distinct from the types of problems that GAs can be applied to. GAs often manipulate vectors of real numbers. I'd like to point out that through the fact that you can have multiple outputs in CGP, you could use CGP to find solutions to standard GA problems. You just feed in some fixed constants, use a GP graph for mathematical functions, and output a big vector of real numbers, and then use those numbers to optimize a very wide range of problems. And this has been tried on in a few instances. There's a couple of papers out there on that. But I think that that's quite an interesting thing to investigate in the future. Other future work, there's a form of CGP called positional CGP, which is a little bit like Janet Clegg's representation in that it's floating point, but it's actually different in various ways. I talk about it a little bit in my paper in Genetic Programming and Evolvable Machines, which came out this year in paper form. Also, Dennis Wilson and I used this technique to find solutions to Atari games. So I think that's definitely worth examining in the future. Trying to remove the length bias in CGP by making all nodes equally likely to be part of the phenotype. I think this could be done with a different form of mutation. You just record how many times that node has been hit by mutation, and you make sure they all are roughly similar. So you go to ones that haven't been hit very often, and you hit those with a mutation. That could be tried. There's been quite a lot of work on parameter choices and algorithms for CGP, and by Kaufman and Kalkreut, and I think that there's more work that needs to be done there. But I remember them finding that simulated annealing works quite well with CGP. So maybe a different algorithm is best. I've mentioned solving GA problems with CGP. You can use the technique I've discussed previously on that. There's lots to do on mutation strategies. Goldman's single mutation strategy works very well. But there are others, and I've recently read some papers suggesting a few others. So that's an active area of research for CGP, a very, very important one. Crossover, well, we concluded that no general form of crossover has been developed that works well for CGP across many problems, so that's definitely an open research area. Once you've got a real valued CGP representation, you can actually apply other methods, other evolutionary methods, highly developed, to the CGP representation. For instance, you could use CMAES. That would be interesting. And I'm currently working on, as I've mentioned earlier, using CGP to evolve the programs inside dendrites and neurons, so it builds a brain-like structure which solves multiple problems. In other words, general AI. There's a lot going on there. So to conclude, Cartesian GP is a graph-based GP method. It's very, very flexible and general, can represent many types of computational structures, from programs, circuits, neural networks, systems of equations, algorithms, and probably many more. It's compact, fast, simple, and easy to implement, and can handle multiple outputs easily. It has a unique form of genetic redundancy, non-coding genes, which make the mutational search highly effective. And the effectiveness of CGP has been compared with many other GP methods, and often it's very highly competitive. So that concludes the tutorial. I hope you enjoyed it.